is a renowned scientist, academician, and researcher in pharmaceutical science and nanomedicine with over 17 years of research expertise. She currently serves as professor in the Department of Pharmacy and holds the esteemed Shri B.K. Birla and Srimati Sarala Birla chair professor position at BIT Pilani, Hyderabad campus. Her research emphasizes the development of advanced nanomedicine and targeted drug delivery system for cancer and infectious diseases, including multifunctional photodynamic, chemophotothermal, and chemophotothermal photodynamic therapeutic approaches. Professor Biswas earned her B Pharma and M Pharma degree from Jadavpur University, Kolkata, and then completed her PhD in pharmaceutical science at Wayne State University in Detroit, Michigan, USA. That was in 2008. And she later served as a postdoctoral research associate at Eastern University, Boston. And currently she serves as an editorial advisory board member of ACS Molecular Pharmaceuticals. In addition, she also serves as managing director of Krishawa Life Sciences Private Limited, her deep tech startup based on pharma cosmeceutical innovations, further reflecting her commitment towards translational innovation and industry academia collaboration. So thank you so much, Professor Biswas, for joining us today, and the stage is all yours now.
Thank you, Ms. Aparna, for this kind introduction. So let me share my slides now.
Yes.
Yeah, is my slides visible?
Yes, going well.
Okay. Okay, so thank you, ACS International team, for giving me, ACS India team for giving me this opportunity. So without further delay, I start with my presentation. So today I will talk about a specific work that we have been doing on stimuli-responsive platinum prodrug photodynamic nanomedicines for cancer therapy, and where we actually do certain strategies for hypoxia alleviation and introduce the reduction sensitivity as well. So coming to so just to give a brief introduction on where we are, basically the BITS Pilani Hyderabad campus where I am working is having 3 different campuses in India, and we are I am situated in Hyderabad, and we have a campus which is having more than 6,200 students and faculties, more than 300 staying together in the campus. So like, in Indian campus, how the academic institutions are built to have researchers in the same place to have more quality time on research. So in our nanomedicine research lab, we are working on various nanoparticles, especially for delivering the drugs and therapeutics and diagnostic agents also. So basically, we are working on various directions for nanomedicine for bacterial infection or nanomedicines for cancer therapy. Here, I will talk mostly about the cancer therapy that we are working. So basically, there are several approaches that can be taken via nanomedicines to deliver the drug. So there is a combination approach which works very well in nano platform, where we can have both the drugs together in the nano formulation. We can also have the two different therapeutic strategies together, like chemotherapy along with the photothermal therapy or photodynamic therapy. So basically chemophotothermal or chemophotodynamic therapy. And in this talk today, I will be talking about the chemophotodynamic therapy. We also talk about various how we can actually deliver the drugs for treating infection. So we have several models, and that actually causes a sustained release of the drug and give the therapeutic benefit. So over the years for cancer, we have developed various nanoparticle formulations using the therapeutic agents such as curcumin or anti-cancer drugs, paclitaxel, doxorubicin, olaparib, docetaxel. Now here actually I'll talk about the platinum-based drug, and which is the platinum prodrug that we will be using. So basically, if you see the serendipitous discovery of cisplatin, which is as interesting story as penicillin. So basically, the scientist from Michigan State University, he was a biophysicist and in the Department of Biophysics. He actually was the man behind development of cisplatin. So he was not a medicinal chemist, not a discovery scientist, or maybe drug delivery scientist. It was a discovery which came accidentally, and it happened because the scientist had a vision, or a curious mind to see what is happening in his research. So he was basically working on this bacteria E. coli, and he was trying to see that when the electrodes are applied, like electrical pulse is applied, then whether that is affecting the growth or not. It was the story in 1978, and he put two electrodes of platinum, and after putting these electrodes and leaving the bacteria overnight in the bacterial culture, and he found that these organisms are basically elongating, and there is it is giving coming to a different shape, but they are not basically growing more, but they are basically stopped the cell division, but they are basically having some elongated structure. And later on, when he analyzed that solution, he found that something like this structure formed, which is actually the cisplatin. As soon as it came into the observation, then the animal study was done, and it has been found that there is a remarkable tumor volume reduction by using this particular platinum drug, and the rest of the things are actually history, because the platinum drug became so potent, and it was used in variety of cancers. And as you can see that now, from 1978, we haven't come very far, but we have three FDA-approved platinum drugs in the market. And we also have several non-FDA approved drugs, and the approved countries are Japan, China, and South Korea, and where you can see that the drugs are getting modified and to introduce some groups. Now, why there is a need for modification is because the platinum is very toxic. So it has several toxicity. Nephrotoxicity is very much, and then there is also neurotoxicity and several GIT related problems, nausea, vomiting. All these problems are there. And if you see the structure of the first compound, like cisplatin, you can see that this is platinum has a tendency to coordinate with many other biological macromolecules, and that way it is getting, forming the adduct with other biomolecules, and that basically, non-specific binding is occurring in the bloodstream, and which is making it difficult, and, as it is highly cytotoxic, so it's basically causing the non-specific toxicity. And so this has been, modified, and as we can see that the oxaliplatin, which came in 2002, has overcome some of these platinum related toxicity found in cisplatin. However, it is not there yet, and therefore there is a need to deliver this drug in a different way, so that we can have some better effect. Now, I will talk a little bit about the nanomedicines, like why we need the nano formulation. Now, basically, if you see the nano formulation, they have a benefit of accumulating to the tumor cells by enhanced permeability and retention effect. Now, basically the concept is when the nanoformulations are systemically administered, due to the complex pathologies in the tumor, the tumor vasculatures are leaky, and that allows anything which is less than 200 nanometers to accumulate in the tumor environment. So there is nothing to be done, but it is going to the tumor environment. And of course, throughout the process, it's also going to liver, kidney, and some of the excretory organs, because our immune cells get activated by having this nano formulation around. But the one good thing is that if we do certain modification on the nanoparticle surface by using the polyethylene glycol coating or any other non-ionic surfactant coating, then it gets unrecognized by the macrophages, so which actually gives us the benefit of long circulation and eventually getting accumulated to the tumor environment by enhanced permeability and retention effect. And that is possible because there is a leakiness in the vasculature, and that happens because the tumor cells are rapidly proliferating, and that requires the food, so blood vessels are immaturely developed. So basically, we have to take care of using the nanomedicines. We actually can overcome many things, like we can overcome the toxicity, we can have the tumor targeting, we can also have the pharmacokinetic property improvement, because the drugs are not directly interacting with the blood component. So it is just the polymer and the drug is embedded inside. So that actually takes care of the toxicity in a significant way. So our proposal, like what I will discuss today, is mainly talking about how we are actually reversing the hypoxia. Now, I want to talk about the hypoxia, which is actually a great limitation for any chemotherapy or radiotherapy. The reason being that when the tumor proliferates, the core of the tumor becomes hypoxic, which is having the less oxygen. And the more tumor grows, the bigger the hypoxic region develops. And we have the cancer stem cells residing there. All the drug resistance develops. And then basically there is a lot of transition happened in the cells, and that allows it to be more robust to the external changes. And those environment doesn't take the radiation therapy well, because the radiation needs the molecular oxygen. Even the chemotherapy also need to go to that area, and the blood vessels are not there. So we are unable to deliver the drugs to that particular region. So we need to have some strategies for hypoxia reversal. Now, we also have a component which is photodynamic therapy. So I will be using a photodynamic component, photosensitizer, also in my rational design. So basically, photosensitizers are light sensitive, and when the light falls on them, they undergoes certain changes, come to the excited state, and when they goes down from the excited state to the stable state, then they basically releases the releases the energy that they have, and that actually causes the production of reactive oxygen species in presence of molecular oxygen. So we can see that the photodynamic therapy, in order to be effective, it also requires the molecular oxygen in the vicinity. So basically, there are a lot of clinically approved photosensitizers which are available, but we can see that two things to be considered here: that the laser that is being applied is in the 600 range, and which is not so near infrared, but it is still better than using UV, the light in the UV range. So these are basically the second generation of the photosensitizers, and our strategy now is to go further to near infrared, go up to 800 nanometers, so that the NIR radiation can have more penetration and less toxicity to the healthy cells. Now, if we can see that these are mainly associated with the superficial cancer, and oral cancer comes in this, and so the photodynamic therapy is very active for the head and neck cancer and for skin cancers. Now, we will talk about chlorine-6, because I'm using this chlorine-6 for development of nanomedicine. If you can see that this chlorine-6 has several carboxylic acid groups, and it has the absorbance at 660 nanometers. So we can actually use this 660 nanometer wavelength to activate this. But however, as you can see the structure, so this is basically having poor aqueous solubility, and and in the bloodstream it aggregates, and when they aggregate, their photosensitizing ability is lost, and they actually have very low uptake by the cells. So, and they have the non-specific toxicity because they are not getting accumulated in the targeted fashion. So now what we did as a preliminary work, we actually prepared the chlorine-6 micelles. So micelles basically is embedding the drug in the core of the micelle, and then it basically encapsulating, as I told you that in order to achieve the EPR effect, you need to have the layer of polyethylene glycol or any other non-ionic surfactant on the surface, non-ionic component on the surface. So pluronic, we have used F127, which is pluronic. Pluronic is very well known for its biocompatibility, biodegradability, and non-immunogenicity. And if we take this F127 and we could actually encapsulate the chlorine-6 inside the core of the micelle, and we can see that, and you can see that we formed a nice spherical structure, and these micelles are well formed, highly stable. And the good thing that it actually increased the solubility of the aggregated chlorine-6. As you can see, if you dissolve them in water, it actually aggregates, and you can see they're precipitating out. But when you have this micellar form, it basically increases, and you can see that even if we increase the concentration also, it's not precipitating, and it's adjusting to that water volume. Basically, we did some of the cytotoxicity analysis, and we used this oral cancer cell line. And as you can see here that, there is a laser, without laser toxicity, but with laser, the toxicity is actually, manifold. So we can actually increase the toxicity by the laser irradiation. Now coming to ROS generation. So basically the photosensitizers are active when they generate the reactive oxygen species. So we should actually see the generation of reactive oxygen species after we administer the photosensitizers, because that is the way they actually kill the cancer cells. There is no other way or other mechanisms that they work. So basically they have a robust increase in the development of all the oxygen related radicals, superoxide or hydroxyl radical, hydrogen peroxide. So all these things increase along with it. So as you can see here that with laser, that this is actually increasing, and we could see a high shift in the flow cytometry, that it is basically working on inducing the reactive oxygen species. Now, why we see that the micelles are giving more reactive oxygen species? Because they are basically in the non-aggregated form, highly dispersive, and they increasing the solubility, and so they are basically more effective compared to administering the free drugs. So similar observations were also found in their other therapeutic efficacy when we actually use them to find out that whether they are disturbing the mitochondria or because the mitochondrial disruption is the first step to induce the apoptosis. So we could see by using a known dye, which is JC1, and we could see more green signal which is coming from the aggregated these cells or aggregated mitochondria, that we could see the effect. Now, in vivo study was also performed on these oral cancer cells, and we could see the reduction in the tumor volume as compared to the free chlorine-6. So, which gives the rationale that we need to, make them dispersible. We need to, because the reason it is important, because right now now in clinic, there is not many, or in clinical trial, there are not many nano formulations available, but they basically have the potential to increase the solubility, reduce the aggregation, and they have the, they make the drug stable in the systemic circulation. They improve the pharmacokinetic properties of the drug. So it is very important that we should actually look into the nanomedicinal formulations. And we can also see that they are basically increasing in the tumor. They basically, having more apoptotic cell death by tunnel assay, and by reactive oxygen species or by proliferative marker. We could see that we could actually stop the proliferation by judging it using the Ki-67. And also another, the effect that we could very well observe, that with these treatments, we actually, reduce the chance of tumor metastasis. So when we use this oral cancer, then there is a chance of getting it accumulated in the lungs, and we could see that there is no metastasis because the formulation was more effective compared to the free formulation. So coming to so with all this concept that we could actually load C6, the next approach that we had taken is a little different way, and we basically here introduced chemo photodynamic therapy. So chemotherapy component was platinum, but here we basically have a conjugated form of platinum. So we did not use the PT2, but in spite of that, we use PT4, and we also had taken the chlorine E6. So basically, now what is like why we want to have a different approach for that? Because we want to deliver oxygen as well in the compartment. Now, why we need the oxygen? As I already told you, that the oxygen is required because the PDT doesn't work without oxygen, right? So these basically, we actually have taken this scaffold. We designed this scaffold. Now, coming to MPEG PLA, so we already have proven that the C6 actually works well with micelles because it reduces the aggregation. Now, what we did here, we actually used the MPEG PLA micelle, and I showed you the structure of chlorine E6, that where there is a 2 free carboxylic acid groups there, actually 3 carboxylic acid groups there. So we actually thought of taking 2 carboxylic acid groups and bind 2 molecules together in both the sides. So the chlorine E6 was acting as a bridge, and one COOH group was bound to PLA, the hydroxyl group of PLA, and the other one was bound with the platinum 4, and the platinum 4 was additionally conjugated to the additionally modified with the azide group. Now, why we need to go to that azide group is because it basically causes the generation of the molecular oxygen. Now, platinum azide, which actually works on the 5' GMP, and then it basically produces the molecular oxygen. So we we try to overcome the limitations of not having the molecular oxygen in the vicinity, because we design the micelle in such a way that this will produce the oxygen as soon as it comes to the tumor microenvironment. Now, how we do this is we have taken this cisplatin, which is dichlorodiamine platinum, and we treat it with the silver nitrate. So we basically transfer this chloride to nitrate, and then we react. We convert it to azide. We treat it with NaN3, so to make it the azide group, because we need this azide for making the platinum azide, which will react further with the 5' GMP. And here that 2 hydroxyl groups are introduced using the hydrogen peroxide, and this hydroxyl group, gave a good opportunity for us to, get the other derivatization done. Now, what we have done here, we took the, chlorine E6, and basically the chlorine E6 was, now this molecule was further treated with chlorine E6. Now, where the conjugation, further conjugation can happen only in these two hydroxyl groups. So we have taken one hydroxyl group occupied with it. The other hydroxyl group was not touched because it might give the steric resistance. So we just have this conjugate, and now we have now this chlorine E6 can have two additional carboxylic acid groups. So one of them were introduced with the further conjugated with the MPEG PLA, and where there is a hydroxyl group which actually reacts with it to form the final conjugate, which is MPEG PLA C6 PT4 azide diazide complex. So that was so this is the structure that we found. And so what are the benefits of those components? As I said, that MPEG will provide long circulation, biocompatibility, and further the EPR effect. PLA will give the biocompatibility, hydrophobicity, and photosensitizer will produce robust ROS generation. And then PT4 azide, basically PT4 diazide, will produce the nitrile radical, azidyl radical, hydroxyl radicals in presence of 5' GMP, and also will produce the reactive, also will produce the molecular oxygen. So together, they can actually eliminate the hypoxia by working in the tumor microenvironment. So all this, keeping all that in mind, so we had done the synthesis successfully, and all the small molecules were synthesized, analyzed by LCMS, and then we found out that they are formation by NMR, HPLC analysis, and also we have done GPC, because MPEG PLA is a polymer, and when we conjugated that MPEG PLA with chlorine E6, platinum as a diazide group, the molecular weight has been increased. So we could find out by jet permeation chromatography, and NMR and IR was performed to find out the stretching and bending by introduction of the new groups. So MPP CPN micelle, which was basically prepared the normal way that the micelles are formed. So we had done the thin film hydration technique, and and finally the micelles were lyophilized. So you could see that we could get spherical sized particles, and the stability of the particles were good in plasma. They were better in stability study, as we can see that at 4 degrees centigrade they had performed good stability. However, it was not as good in the room temperature that we have found. Now, coming to certain benefits of photodynamic therapy, many times you will see that the photodynamic therapy is also associated little bit rise in the temperature as well. So, basically, we also studied that whether it is having any photothermal effect, and we could actually see that there is a little uplifting of a little increase in the temperature. So it was around a little more than 45 degrees centigrade was observed with this. Now, this is one good, the significant milestone of our study, that we could see that it is actually producing the molecular oxygen, and we could determine by using the oxidation green, which is a hydrogen peroxide sensor, and in presence of our formulation, it is basically, producing the molecular oxygen. So we could, like after forming this, we, as a procedure to go to in vitro study, we do the cytotoxicity analysis, and with laser and without laser. The laser being used here is 666 nanometer laser, and we could see that there is a cold toxicity However, the laser-mediated toxicity was more compared to the without laser toxicity. The nuclear staining and DNA fragmentation is very important to analyze, because we need to see that whether the platinum is giving the better effect and how that adduct is forming. So we could actually see that there is a DNA damage which is observed by by SDS page, and also we could see that this nuclear staining actually proves that basically there is a fragmented DNA component present. So we could also see that the ROS generation is actually getting interlinked with the mitochondrial membrane potential downregulation as well. So we could see that the disturbed mitochondrial membrane, and which is actually triggering the next phase of apoptosis. So live dead cell assay, basically, this is also a preliminary assay to see that the cells are dying or not. So what is basically happening with the cells? So we could see a lot of dead populations, and we could actually see that, when we use this platinum, but compared to that, actually, we could see a lot of cell death in our formulation, and that is envisioned because we have both the components there, like chlorine 6 and platinum as well. So we also mixed that chlorine 6 and PT2 together, and we could not see the similar benefit of using our formulation, because here it is not just PT2, it is basically PT4. Now, why we want to give the PT4 is because it is PT4 is more structurally stable. They can form better DNA adduct also, and they have actually minimized the non-specific bindings. PT4 is not in the clinic yet, but there are a lot of investigations which are happening. If there is the PT4 derivatives can be prepared. So cell cycle analysis also was performed, and you can see that there is a complete shift, because we see that how the C6 is interacting, or how the platinum, it was a dual effect which was observed, and we could see that, yeah, definitely the cell cycle is getting arrested drastically when we use our formulation. So we actually in our lab we are making the spheroids in a routine manner, because we think that the spheroids are better model to analyze tumor progression, or analyze the effectivity of the formulation, because the monolayers may not give the whole picture. So here we made the oral cancer spheroid, and we could see that the nanoparticles are actually getting penetrated and also giving effect. Now, of course, the hypoxic core is not, that there is a reversal in the hypoxic core, and which is also causing the penetration of these the formulations, and also the effectivity of the PDT. So here we have developed the human FADU oral cancer, and what we see here that with the formulation, this is more effective compared to all other tested formulations. So we have used that chlorine 6, and we have used this chlorine 6 PT adduct, but we could not see that effect which we are getting through in our micelle. And that has made sense, because it is a nanomicellar formulation, and we anticipate that it will be delivered to the tumor environment very well. And also there is no oxygen component given. So the formulation which is delivering that three therapeutic strategy will be more effective. So we could also see by the tumor section that the hypoxia actually was reversed. So we had taken the HIF-1 alpha. So basically, this antibody, anti-HIF-1 and alpha antibody was used, and so the main, the hypoxia markers are HIF-1 alpha or HIF-1. So basically, we see the down regulation of that, and also we have taken the CD44. CD44 is basically, will tell us that how the endothelial cells are working in that, because when the hypoxia is occurring, then there will be abnormal endothelial cells growth, but when there is oxygen delivery, then that gets minimized, and there will be more structured blood vessels which are forming, and which is also indicated by CD upregulation of the, downregulation of the CD31. So HIV1 alpha got downregulated, so we could deliver the oxygen, and also CD31 expression was low. So with these, basically, we conclude that this is very important to make the formulations multifunctional, so that we can address the whole situation and find out that, like, one drug may not be enough, but when we have the multifunctional approach, then we could actually do better job. Now, then also platinum, PT2, cisplatin, oxaliplatin, they have various toxicity, nephrotoxicity, then GIT related disorders. But if you make the platinum 4, then it is not, you are blocking the site, so it's not going to react with the biological macromolecules. So that is wonderful opportunity to deliver the drug to the target, because it is not going to get distributed through the whole body. And also, if we have the light activated therapy, which is an adjuvant of the chemotherapy, that is going to have better success, because the two drugs are not competing with the pathway, but it is basically, causing the apoptosis, and further with the further necrosis also, because there is a upsurge in the reactive oxygen species. And here we have also shown that delivering oxygen is so essential for making the photodynamic therapy effective. So with this observation, we have also done some other studies with the platinum. So I'll mostly talk about our study, what we have done for platinum today. So here we have taken another approach, so which is like we are using human serum albumin. Now, many of us know that human serum albumin is, a great transporter protein, and this basically used to transport many lipids, vitamins, many other, components, to different cells. And the reason we use is because there are a lot of very well-known drug nano formulation in the clinic, which is paclitaxel bound this albumin, which is Abraxane. Now, after the great success of Abraxane, we have found that the potential of using human serum albumin is manifold, because it is very, very stable. The half-life of human serum albumin is almost 19 days, and it is basically, it has both the pockets, hydrophilic and hydrophobic. So it can actually load, even though it is water-soluble protein, but it can actually load hydrophobic drugs also, so which is a great thing. So now, another major advantage for human serum albumin is that it basically has a tumor targeting potential. Now, how does it target to tumor? Is by two mechanisms. One is there is a protein called SPARC, and which is overexpressed or abundant in the tumor microenvironment. And there is also GP60, which is present, which is a protein present in the endothelial cells, and also in the on the cancer cells. So this basically GP60 and SPARC, these two overexpression causes the nanoparticles to eventually accumulate in the tumor environment by active targeting process. So passive targeting will work if the nanoparticles are less than 200 nanometers, but active targeting will also be, active in this particular case when there is a albumin nanoparticle. Now, here we have done certain other strategy that we have actually taken the reduction sensitivity by, relying on the upregulated glutathione in the tumor environment. Now, this is another well-explored concept. Basically, that in tumor environment, the level of glutathione basically increases to more than 100 to 1,000 fold compared to the normal cells. Now, what they are basically doing, if we have certain formulation which is having the thiol group, we can basically introduce these thiol groups, and this GSH will be engaged, and then we will have and also the bond can be broken by the reduction sensitivity. Now, what is our strategy here? So what we have done here, that we basically made that the PT4 conjugate, and that we made bound with, so we had that bound with the human serum albumin. Now, in order to make it bind with the human serum albumin, we first took the oxaliplatin, and then by using the process that I showed before, we used the hydrogen peroxide to introduce the two hydroxyl groups here. And then we had used one disulfide linker to link with the human serum albumin. So we have used this diglycolic acid as a linker, and it established the ester bond. And then we have that one COOH group open, which basically is reacting with the human serum albumin and making the amide coupling over there. So we are actually introducing stimuli-sensitive linkage between the human serum albumin and oxaliplatin, which is PT2. So, and then what was the another strategy that we had taken? We basically used two cross-linkers to further, we took two cross-linkers to cross-link the human serum albumin nanoparticles. Like to form the nanoparticles, we had taken another cross-linker. So basically, human serum albumin conjugated to PT4 conjugate was taken, and that was undergone by the dissolution technique. And while making the dissolution technique, we used two cross-linkers, one without disulfide bonds, the other one with disulfide bonds. So the back was with disulfide bond, and glutaraldehyde, which doesn't have the disulfide bond, but which can also cross-link the human serum albumin very well to form the nanoparticle structure. So these two, the cross-linkers were used because we wanted to see the effect of, dual reduction sensitivity for these OSHB nanoparticles compared to the OSHG nanoparticles. So G stands for glutaraldehyde, used as a cross-linker, and B is back is used as a cross-linker. So as we already introduced disulfide bond in PT4 complex, and which is bound to HSA, and we also introducing a cross-linker with the disulfide bond. So we are introducing the dual reduction sensitivity. And so after making the formulation by the dissolution technique, which uses ethanol, and we could actually successfully prepare the nanoparticles after the lyophilization. Here, glutaraldehyde is, very commonly used to prepare the nanoparticles, HSA nanoparticles, but BAC was introduced, and so it, the reason being only that it has a reduction sensitivity. So after preparing this, we could see that the nanoparticle basically formed because with the GPC we could see the higher retention time for oxases HSA because of the introduction of many other components with the human serum albumin, and also the small molecule by using the dithiodide glycolic acid conjugation basically works, and we could get the desired peaks. The nano formulations were basically nano size that we could observe, and moreover the human serum albumin also need to be stable. So we could see that this is basically the human serum albumin peak under CD, but we could see that the peak is little shifted because we could say that the alpha helical structure is little bit compromised, but it is not drastically changed to something else. So it is making it a little more restricted. And we could also see the presence of human serum albumin peak in SDS page, and also we could see now we have to actually see that whether the glutathione is really reducing it or not. So we could actually we could do the experiment in presence of glutathione. We could see the effect is very prominent that we could see here. So we could basically see that here only the 20%, less than 20% formulations are released, but here we can see that more than 40% is released because the GSH is basically reducing and liberating the drug out. So we could get that effect. So further, we have done some studies. We could see little change in all the experimental conditions that somehow it is working. But I would also tell you that the monolayer doesn't represent that actual environment because it is not showing that the exact picture of how much elevated glutathiones will be there in the system. So we could actually see the effect which is like here basically we are talking about the cellular uptake, and you can see that both nanoparticles are basically being taken up better way. Like you can see that compared to 1 hour, it is more in 4 hours, and that is due to the fact that it is getting mediated by the GP60 mediated delivery. Now, whether it is really going through GP60, so we have done some studies, and this is one of the study that used chlorpromazine, amiloride or beta cyclodextrin as an inhibitor. So chlorpromazine inhibits the clathrin mediated endocytosis. Amiloride is macropinocytosis inhibitor, but beta cyclodextrin is something which basically inhibits the lipid rafts mediated. So it's basically caveolin mediated endocytosis, and all the GP60 receptors which are localized in the caveolin region. So that we could found that we have found that this is when we put this inhibitor, this is basically completely blocking. I mean, this blocking is much more than other blocks. So it basically says that this this our formulation is getting taken up is by the GP60 receptors. So further, we had also done the in vitro study using the cell lines, and we could see that there are two different types of murine, and the mammalian cells were used, human breast cancer cell lines were used, and we could see that the decrease in the cell viability. And if you see that the cell viability decrease was more in case of DAC, because there is the reduction sensitive component added to it. So we could see that in the in vitro assays of cell viability, we could see always the viability is low, and this study was done in NIH-CT3 with cells which are normal cell, and we could see that there is not much effect, that not much cell killing effect which is observed. So that indicates that we could actually prevent the drug from being cytotoxic to the normal cells. So we could also do some intracellular ROS scavenging, ROS generation assay, and also the effect was similar, as we can see that they basically having more, fluorescence indicating the more, formation of the system. So other assays were also carried out for the DNA damage and or in oral cancer. We also did the in vivo study, and as you can see that there is a reduction in the nanoparticle. There is reduction in the tumor volume using the nanoparticles, and so the other activities were also related. So one of the major use of this is to reduce the toxicity. So we could see that the free oxaliplatin has a lot of nephrotoxicity, and which was not observed when we had the nanoparticle formulation. So that actually comes to the end of my talk. So basically, as I said before, that it is very important to have the nanomedicines for various reasons, and stimuli-responsive nano platforms can be introduced for better therapeutic outcome, reducing the toxicity, improve the pharmacokinetic potential of the drug itself. And there are various strategies that we can take. Also, oxygenation is one of the important thing. If we can oxygenate the tumor, then there is a lot of effect can be enhanced, chemotherapeutic effect, radiation effect. So hyperparation, like oxygen giving, is another strategy. So there is also a lot of catalase that are giving in order to produce oxygen. So we can also consider, using the oxygen incorporated in the nanoparticles. So with this, I would come to the last slide of acknowledging my driving force for this, all the studies. And this is a little old picture. I don't have a very recent picture, but yeah, so these all the students which help in these studies, for doing these studies, and also the main people are here, and also the funding agency. I will say that we are generously funded by DST, ICMR, all the major funding agencies from India. Thank you to all of them, and